Hello guys, welcome to the basic mantra writing classes. We are going to learn about karyotypes. So the number and visual appearance of the chromosome in the cell nuclei of an organism or a species. Okay, so how chromosome is looking in the nuclei is called karyotypes. We can see in the picture the different numbers of chromosome. You can see here also the XY chromosome. Okay, in human there is 22 pair of X chromosome and one is X and Y chromosome, right? So in the chromosome there is short arm and the long arm, right? And it contains a centromere. This central part is called a centromere, right? So there is a four types of staining technique: Q, G, NOR, and C, centromere, right? So in the metacentric, these are X-shaped chromosome with the centromere in the middle, so that the two arms of the chromosome are almost equal, right? So in this case, the centromere is in the center, right? In the submetacentric position, if arms lengths are unequal, the chromosome is said to be submetacentric. So centromere not in the center but little bit above or little bit lower. Then it is called submetacentric. Acrocentric is when the centromere is near the P arm. Okay, near the P arm is acrocentric. What is telocentric? When it is centromere is on the end side, right? Centromere on the end side is called centromere. Telocentric, okay. Now you can see in the picture metacentric chromosome. Metacentric chromosome. So in metacentric chromosome you can see here centromere is in the middle. In the submetacentric, centromere is away from the little bit away from the middle, and in acrocentric it is far away from the center, right? And in the telocentric it is on the end side, right? A chromosome banding pattern is comprised of alternating light and dark strips, or bands that appear along its length after being stained with dye. A unique banding pattern is used to identify each chromosome and to diagnose chromosomal aberrations, chromosomal breakage, loss, and duplicated or inverted segment, right? So the study of karyotype is made possible by staining. You can see chromosome by staining. So usually a suitable dye is gimsa is applied after cells have been arrested during cell division. When cell is arrested during cell division, then this will be applied. First colchicine is applied and then proteinization, and then gimsa stain, right? So what is most preferred cell used in the study of chromosome is white blood cell, WBC. Okay, WBC is used because it easily get divide and grow in a tissue culture. Easily divide and grow in a tissue culture. Okay, sometimes observation may be made on a non-dividing interface cell. Sometimes interface cells also has been taken, right? So to check the sex of unborn fetus, we use gimsa staining. Okay, we can use WBC. Okay, unborn fetus can be determined by observation of interface cells. See amniotic centesis and bar body. So here you can see WBC cells is taken from the body. Then phytohemoglutinin is applied and then put it in a tissue culture for 3 days, and then colchicine is applied and put it in a hypotonic saline solution. Then cell get fixed and put it dropwise on the glass side, and then put it trypsin for proteinization, then gimsa stain, then analyze metaphase spread, and then you can see this picture in a microscope and they take out picture, cut chromosome series wise and put it on a paper, and that is called karyotype, right? I hope you understand this karyotyping and gimsa staining. So what is gimsa staining? In G banding is obtained with gimsa stain following deletion of a chromosome with trypsin. So what we have discussed in the picture, that is called gimsa staining. In this, basically 80 rich region get stained. Okay, and it is called heterochromatic region. And in the reverse bending, reverse is opposite of gimsa. So if 80 rich region is stained in gimsa, then in this GC rich region stained in our bending, right? You can see clear difference. Here 80 rich and here is GC rich. Dark region is GC rich. In C banding it is used for the centromere, to color centromere, right? You can see here the darkest region is 80. Darkest region is 80. In R bending, darkest region is GC. And in C bending, there is a darkest region is centromere, right? Centromere. This is called C banding, right? Okay. In Q banding, it is fluorescent pattern obtained, right? So both 18 GC's reason get stained, but 80 get more stain, right? It more fluoresces. It is basically for 80 rich reason. So bind both, but only 80 quinacrine complex fluoresce, right? So only 80 rich reason will glow more. In T banding pattern, it is used for the visualization of a telomerase. In silver staining, to see rRNA genes within the nucleolar region, silver staining has been used. Okay, this is staining. Now we can see disorder. Why chromosomal disorder happen? It is happen because of non-disjunction. It means there is no proper separation of a parental cell. Okay, so parental cell not properly get separated. Due to this, one extra chromosome arise with the one pair of a chromosome. In deletion, some part of chromosome get deleted. Okay, if there is A, B, C, D, so C, D get deleted. In duplication, A, B get duplicate. So A, B and A, B chromosome, extra chromosome copy found in duplication. In translocation, some part get breaks and attaches into another chromosome. That is called translocation. So in Turner syndrome, what happened? There is a missing of one chromosome. In Klinefelter syndrome, there is one extra X chromosome. Okay, so XY is male, right? In male, X character is extra shown. So in this condition, male gets female character. That is called Klinefelter syndrome. In Edwards syndrome, 18 chromosome get trisomy. It means there is triple X in Edward on chromosome number 18. In Down syndrome, trisomy of chromosome number 21. In Patau syndrome, trisomy of chromosome number 13. You can see here. In Patau syndrome, there is a cut on the mouth. In Patau syndrome, cut on mouth. In Edwards syndrome, you can see malformed ears, right? In the Patau syndrome, there is extra finger also. Okay, and in this, there is a gap in fingers in Down syndrome, right? In Klinefelter syndrome, in male, there is formation of female character. Okay, so this is called chromosomal aberration. If you like this video, then please like and subscribe this video, right? Okay, thank you so much for this class.